to really take us through an understanding of broadly neutralizing antibodies. Thank you very much for waking this early, Gail, and over to you.
You are most welcome. Hi, everyone. It is still dark in Seattle. The sun has not yet come up. And I noticed Barbara said she's in very snowy New York. I'm in very rainy Seattle, but it's a pleasure to be with everybody. And so just to be clear, this will be kind of an overview of broadly neutralizing antibodies. We won't be diving into the results from the AMP studies today. We'll save that for a future call. And I wanted to start by pointing out this crazy acronym, because everywhere you see it, you see different letters capitalized and different letters in lowercase. And the short answer is that there's no agreement across the field. All of the different scientific journals do it differently. Pretty much any way you want to spell it, uppercase, lowercase, it will probably be correct somewhere. At the HVTN and HPTN, who have been doing the AMP studies, we have chosen to only capitalize the A for antibodies and to leave the broadly neutralizing lowercase. But you'll probably see different variations on that everywhere you look, and none of them are right or wrong. They're all just different. So let's dive in. First off, what is an antibody, generally speaking, before we even get into broadly neutralizing? So antibodies are proteins. They are made in our B cells, which is part of our immune response. Typically, they start being made about 2 days after some foreign thing has entered our body, but they can really take up to 2 weeks to be fully at full strength. They are shaped like the letter Y. They have kind of 2 arms at the top and a foot at the bottom. They are able to prevent infections. They can block viruses and bacteria from being able to attach to our cells to cause an infection. They also are unique because they have memory. And so they will remember the different kinds of things that you have been exposed to in your lifetime. So if you had a cold or the flu last winter and you get exposed to that same cold virus or flu virus next winter, they will remember that they have seen that before. Typically, the antibodies that we make naturally don't last very long in the body, usually less than 2 months. And so we're sort of constantly making new ones. But the ones that are manufactured and given as a medication can be engineered to last longer. And we talk about this as having a longer half-life, that they last longer in the body. So what is it that antibodies do? They actually work in 3 different ways. They can neutralize, opsonize, and sensitize. And we're going to focus today on the ones that do neutralizing, which is what bNAbs do. And just to say briefly what the others are, opsonization is the idea that after the antibody has used its arms to bind onto the virus and block its attachment, it uses its foot to pull the virus into other cells that can kill this. And we refer to this as a metaphor as buttering the toast. The antibody is making that virus look really tasty. So the cells that do the killing are more likely to want to kill it because it looks more appealing. It's now got something really yummy and flavorful attached to it in that antibody. And then the third function is to sensitize. And we talk about this, if you've ever watched an old gangster movie, you'll know that every good killer needs a hitman, or excuse me, every good hitman needs a lookout. And so just the fact that the antibody attaches to a virus serves as a way of alerting the immune system and allowing other cells that have killing function to come along and be more effective killers. But for today, we're going to focus on neutralizing. And what does that look like? So I'm going to let this little animation play here. This is you at the bottom in purple. You're the host, and your cells have these receptors on the surface. And the antibodies are floating around in your blood system. And when a virus comes along, they attach to it, and you can see that they block it from being able to attach onto those receptors. I'm going to let that play one more time through just so everybody gets to see the whole thing again. So the antibodies are just hanging out. They're floating around in our blood. And when a virus comes along that would ordinarily attach to those receptors and cause an infection, the antibodies are able to cover up the parts where that binding would happen. So thinking about, that's what antibodies do generally speaking. So what is a broadly neutralizing antibody? And a BNAB really means it's an antibody that has a specialized function that allows it to block all different kinds of the global strains of HIV. And this is referred to as the breadth. So it recognizes the clade C strains that circulate in Africa and the clade B strains that circulate in the Americas, and so on and so on. Another term that you'll hear that's related is the term monoclonal antibodies. And if we break down that word just a little bit, a monoclonal means that it is a copy or a clone of one single antibody. So one in Latin is mono. So a monoclonal antibody is just one unique antibody that has been manufactured in a lab rather than being a naturally occurring antibody. So a monoclonal is the noun, is what it is, but its function is to be broadly neutralizing. So why do we care? Why do we care that it can neutralize these global strains? Well, we remember that HIV looks very different all around the globe, and that there are many different strains that circulate around the world. And these strains are always mutating. The virus is always, every time it makes a copy of itself, it makes like a spelling mistake. And so we get these variations all around the world. And in fact, the different strains of HIV are different by roughly 10 to 30%. So to think about why that percentage is so significant, think about the people that you know, people in your life, people in this, if you were to look at just the photographs of people who are gathered here today, you would see people that have different hair, different color eyes, different shaped bodies. Some are thinner, some are thicker, some are taller, some are shorter. We have different genders, we have different sex assigned at birth. All of these different variations that we can see with the eye, human beings only vary by about 0.1%. So HIV is different by 1,000 times or more than human beings. So this global diversity is one of our biggest challenges that we're always trying to overcome. And so the fact that these broadly neutralizing antibodies can work across all of these different global strains is really significant. So where do the BNABs attach to HIV? And you'll probably be hearing more about this in the coming years now that we have the AMP results. So there are lots of places where antibodies attach. And I've tried to show the different locations as well as the names of some of the different antibodies. And I have to say, the scientists are not terribly creative. These all have god-awful, horrible names, numbers and letters that don't mean anything to most of us. You can see that the one that we've been hearing about this meeting from the AMP study is VRC01. I've circled it over on the left. It attaches to the CD4 binding site, which is where the virus would ordinarily attach to our CD4 T cells. But in addition to that binding site, there are these other locations as well. So part of what we're going to be hearing about is using different combinations of antibodies that perhaps bind to different regions. So to give you a sense of what that looks like, I want to take you to an example that's from our COVID experience currently, because I think that this particular photograph gives you a useful idea of what those different binding sites look like. So in this enlargement of the view from under a microscope, that's a coronavirus on the left in purple. And kind of in the center of the screen, you can see the protein spike on the surface here in sort of purple and pink and yellow. And you can see that these antibodies, here's the little Y-shaped proteins shown in purple and green. And you can see that they attach to all different sides of this protein spike. And as a result, they're able to block that protein from being able to latch on to this receptor on the human cell. And so in exactly the same way, HIV has its protein spikes on its surface, and antibodies can attach on all different sides, all different features on this spike to help block its attachment. And I see Jessica posted, that's a great picture. I agree. This is from our colleagues at the Vaccine Research Center at the NIH. I don't know that the colors are real. I think that might be a trick of photography, Jacinda. So thinking about this, that these BNAbs attach to this three-dimensional structure. So, oops, I forgot to point that out. So you'll notice that this protein spike is not smooth, right? It looks sort of rough and bumpy. It's got a texture to the surface. And that actually is part of what makes these BNAbs unique. The BNAbs actually recognize this three-dimensional, rough, bumpy surface, whereas ordinarily other BNAbs recognize little, small parts. So I'm sort of showing here a representation of the GP120 protein. And you can see on the right, GP120 is sort of the tip of that protein spike. It's got three parts. And they're this tangle, and I'll show you what it looks like in a moment. Ordinarily, antibodies recognize just little short, sort of straight line pieces of protein in that tangle. Where BNAbs are different is that they recognize this three-dimensional shape. And the way that I explain that to people is by using my fist. If you were to make a fist right now and look at your own hand, you can think of those ordinary little straight line called linear epitopes as like the distance from one knuckle to the next knuckle, right? It's a little straight line, flat spot. But the BNAbs recognize this bumpy three-dimensional surface. So thinking about like where your fingers and your thumb all come together. So that Y-shaped protein with its little two arms can come along and recognize that bumpy, rough surface and attach on to that whole three-dimensional shape. So then we get into some of the vocabulary that makes this science so challenging. And this is some data that actually came about before the AMP studies. This was part of what informed moving forward with AMP. And it shows us this idea of IC50 and IC80. What does that actually mean? So the IC stands for inhibitory concentration. And this is the idea that you need to have a certain amount of the antibody present in the body, in the blood, in order to neutralize 50% or 80% of the viruses that it comes across. So an IC50 means that it's going to take a certain amount of antibody to block 50% of the viruses, or IC80, a certain amount of the antibody to block 80% of the viruses. So again, the numbers change because it's how much virus can be neutralized. And what we learned from test tube experiments in the lab, known as in vitro experiments, was that when we compared the one broadly neutralizing antibody, VRC01 on the left, with another sort of ordinary antibody, B12, we saw these really impressive results. And so kind of here in the middle of the table is IC50, and the concentration of antibody needed was less than 50 micrograms per milliliter of blood. And then here on the right side of the table is IC80 at less than 1 microgram per milliliter. And as the totals show us, the VRC01 was much more effective at neutralizing the antibody at either concentration than a regular antibody was. But it was also variable. It didn't work the same against all the different clades. So it worked a little better against some clades than others. And, but overall still much more effective at neutralizing. So when I look at diagrams like this, I pay attention even just to the colors. So you can see, just looking on these, in the tree diagram here, you can see the different clades are shown. Here's clade A, here's clade G, B at the upper left, C on the right. And so just by looking at the lines, each line represents a different sample of HIV that was tested. There were 190 different samples altogether. And just comparing the colors, you can see by the amount of red how much more effective VRC01 was at neutralizing HIV. So this gives us an idea about how we understand these antibodies going forward, and paying attention to just how potent they are, how much antibody is needed. And that potency can be graphed like this. So each of these different colored lines is a different broadly neutralizing antibody. And I've shown the names here. And Daisy just asked if this is in the lab. And Daisy, that's correct. Those were all tests done in a lab, in a tube.
Thank you.
So this graph is showing the idea of just how potent, how strong are each of these different antibodies. And what we're looking at is on the vertical axis, how much neutralization was happening. How strong is it? And on the horizontal axis, we're looking at this inhibitory concentration. So how much antibody did you need to achieve that level of blocking? And so the idea to make sense of this is that you want to sort of look, where's the peak of the curve? And the further to the left that the peak is, the more desirable it is, because that means that you could use a smaller dose of antibody to achieve the most neutralization. So you can see kind of right here, sort of in the middle, is where we start to see these peaks happening, but they continue to grow. So perhaps right around here, a little over 1 microgram per mil is where we see the peak for TENI8 in the tan line, for example. VRC01 was a little less potent. 3BNC, a little less potent yet, and so on and so on. So we're looking at kind of this middle region here to say, okay, that seems like the point where we get the right amount of the dose of the antibody to achieve the most amount of killing or blocking. And that's the idea of the potency. So the perfect BNAb is going to be broad. It's going to work against the widest number of strains, but it's also going to be potent so that we can use the smallest dose and be really efficient, but achieve the best amount of blocking. So that is the end of my overview, and it brings us to the next part of the program with my colleagues from AVAC take it away. Let's see, I see a question. Barbara asked if IC50 and EC50. I haven't actually seen EC50, so I'm not 100% certain what that even stands for. And if anyone knows and can tell me, that would be great. Effective concentration, I don't know if those are the same, Barbara. I'm not sure how effective concentration is defined. So I'm not 100% certain whether those are the same. The other term that you sometimes will see is ID50 or ID80, which is the inhibitory dilution. And that refers to the dose of antibody. The dose is given based on a person's weight. So for example, in the AMP study, we tested a low dose and a high dose, 10 milligrams per kilogram or 30 milligrams per kilogram. And so how diluted was the antibody based on the size of the human being. So depending on how effective concentration is defined, it might overlap with one of those terms, or it may be something completely unique. I'm uncertain. So Manju's just put a definition in the chat for us. I'll have to sort of ponder on that and think, maybe while this is unfolding, I can think on that a little further. Go ahead, Daisy.
No. It's actually, we're going to hand it over to Stacy.
Okay.
Yes, Stacy takes over with the question. Thank you very much, Gail. That was helpful. And we see other questions coming through. And please bring more and more questions, and so that we prevent the confusion, if any, as we had mentioned.
And remember, there's a quiz coming up. So pay attention.
Great. Yeah, I mean, I think we just wanted to open the floor to questions from participants. I know, but first of all, just Gail, to say thank you so much for that overview. I know there were a few things that that just hearing you explain it, because I know you've been doing this for a long time, really kind of helped me understand better. I think the IC50 and the IC80 especially, and I know we're not going to delve into the AMP results themselves specifically in this session, but I think that is something that is just, we constantly see being linked to the results and linked to potentially the effectiveness or the efficacy of a BNAB or of a BNAB product. So I think that's something to me that's really important to understand, but has been and as Daisy mentioned at the beginning, I've been trying to wrap my head around BNABs and AMP and VRC01 and the results for a while. And I think you just have to keep chipping away at it. So that was super helpful.
And I think, it's useful. I'm looking at this again, the definition that Manju posted in the chat. And thank you, Manju, because that's helpful. Again, remembering the IC inhibitory concentration, right? So how much BNAB did you need to inhibit the virus? And then the number is telling you the percentage of virus that was inhibited. Mm-hmm. So did it inhibit 50% of viruses? Did it inhibit 80% of viruses? I think that's useful to keep remembering. Sometimes the acronyms are useful, but sometimes they get in the way of remembering what it actually means. So I think it's useful. I'm looking, Jessica asked, what do you think are the key things we need to know or ask about when we see the results from lab studies? So I think, what AMP showed us is that once again, the human body is a more complex thing than just what we see in a test tube. So while we had, excuse me, these great results in a test tube, it was a little less shiny in actual human beings. But I think, again, it's for me, the thing that's really hopeful coming out of AMP is that it really gave us a very clear direction of the way forward. And so if you only were to take away one thing from the AMP results, we proved the concept. An antibody can be used to prevent HIV, but what we saw is one antibody is not enough. It's not broad enough, it's not strong enough. And so what we need to look at going forward is finding the best combinations of antibodies. And for me, the most useful metaphor to compare that to is what we've been seeing in HIV treatment since the 1990s, right? And we learned that AZT was a terrific drug, but monotherapy wasn't so great. And we needed to have combination therapy to really be effective at treating HIV. And I think that this result gives us a really direct parallel. One antibody, yes, we prove the concept that it works, but monotherapy or monoprevention in this case would not be sufficient. And so to me, it's those kinds of things. When you see the lab data, you have to sort of ask yourself, okay, well, what were they actually using? Were they using one antibody, a combination? What kind of, was it an IC50, an IC80? Obviously, if you can block 80% of viruses, that's better than 50%. So those are the kinds of details to look at. Barbara asked about how this was explained to participants. So Barbara, I should tell you that I took my slides today from presentations that we made to go with the informed consent process in the AMP study. So these same kind of slides that I used today are what was used with participants who were or potential participants who were going through an informed consent process and were deciding whether they might want to join the study. And I think from what I've heard from community educators, and actually I'll look to Ntando because he was one of those community educators. I think the little video animation is probably one of the most effective pieces that we developed to explain antibodies. For people to actually see, and Ntando's nodding, okay, that's good to know. For people to actually see how an antibody, they're sort of present, and then when the virus comes along, they're able to attach and block the virus from being able to bind onto cells. I think that was probably the most helpful piece for most people. Looking to see.
Can I jump in, Gail, and ask, just building on that, because were there particular kind of concepts or issues that you felt were kind of difficult in terms of the community engagement, or yeah, maybe just share some of that?
Yeah, so let me tell you what we thought was going to be difficult and how we were wrong. What we thought was going to be difficult was convincing people to come in and get an IV infusion, or in many of the African communities, this is known as getting a drip. And most people associate getting an IV or a drip as some sort of treatment that you get if you're very ill. And it's something you have to go into a doctor's office, into the clinic to get. It's not something you can do by yourself at home. And so most people associate it with being sick and needing some sort of treatment. And we were concerned that would be perhaps stigmatizing, or that people would be confused that perhaps we were giving them HIV, and that was why they needed to have an infusion, was to make it go away. We had a lot of concern about whether we could actually convince people to give this a try in the clinical trial. And what we found was that was not actually a concern at all. We have had over 94% retention among participants. Over 40,000 IV drips have been given. And the people have been very willing. It's not painful. It takes usually a half an hour or less. it's very hydrating. So you come away kind of feeling good because now you're nicely hydrated. So it's actually been something that we thought would be a problem, and we were as wrong could be. It's actually gone very smoothly.
I never knew that about the hydration issue. That's really cool.
If you think about, when you've seen people like these ultimate athletes, like people who run a marathon and, so they're sweating a lot and they get really dehydrated, the first thing that they do, you see, the runner falls down exhausted, right? And one of the first things they do is give them an IV of
fluids in there. Yeah.
Right? Just building your fluids. So yeah, so when you're done, you feel pretty good.
Yeah.
Your skin looks great,
So great. So we have a little interactive multiple choice thing just to kind of continue stimulating conversation. I did have one question to ask for you, Gail, and this does link a little bit to kind of some of the things that we're hearing about the results. So feel free to kind of just mix. But I think some of the issues that we're hearing about is this issue between sensitive viruses and resistant viruses. You don't need to kind of go into what that means in terms of AMP results, but I think one of the things that has been really confusing to me when I am hearing the results presented and hearing some scientists sort of pick apart the results is a lot of discussion around resistance. And I'm wondering if there's a way to distinguish resistance of a virus to a BNAb versus like ARV resistance, because I feel like that is that's a challenge as we're trying to sort of think through, especially as advocates and community members.
Yeah. So it's actually, the ideas are actually quite parallel. So I think what AMP showed us is that success is all about the virus. And if this virus that you happen to get exposed to is one of the ones that's sensitive, or I might use the word vulnerable to an antibody, then we're successful. Great. But not every virus is vulnerable to every antibody. And so, and you can think about it, if any of you have ever had an illness where you had to take an antibiotic medication, you probably know that there's lots of different antibiotics, right? But you might use antibiotic A against this infection and antibiotic B against that infection. Or maybe you're somebody who has an allergy, so you are sensitive to that medicine. And so you have to use a different medicine, right? And we see that with HIV treatment, right? You might take a treatment for some period of time, and over time you it becomes less successful for you. So that's the idea of resistance as well. So it's really exactly the same kind of idea. I'll use myself as an example. I am allergic to the kind of antibiotics called sulfa drugs. And they give me a terrible allergic reaction. I get itchy bumps all over my body. It's miserable. So if I get sick and the doctor needs to give me an antibiotic, he already knows that I'm sensitive to sulfa drugs and I need to be prescribed a different kind of antibiotic that's going to work for me, that's going to be more successful. So it's the same idea. We have, and one of the results that came out of AMP is that we now know that there's a lab test that we can do that will predict which viruses are going to be susceptible to which antibodies, and looking at for that match. So if we know which viruses are circulating in a particular area, I showed you that map, and we know that there's different viruses circulating in different regions, right? So if we can look at which viruses are circulating, we can predict which antibodies are going to be most successful. And we can use that information to guide how we design the clinical trial and try to save some time by putting forward the best combination that we think, if we're going to do this study in sub-Saharan Africa, we should use these antibodies, and because we know that these are the viruses that are circulating, and we can be more targeted to each region in that way. Let's see, I see
Great.
Jim.
Thanks, Gail. Jim
raised a question. What's the thinking about types of products? Yeah. So Jim, this is sort of the $1 million question, right? What is actually the future for a product that could be brought into the marketplace and made available? And the thinking is really, it's really diverse. One of the things that's been talked about quite a bit is the idea of being able to use these antibodies for prevention of mother-to-child transmission. You could give IV infusions, for example, to a pregnant person to prevent transmission to the fetus while it's in utero. And then you could continue to give both mom and baby antibodies post-delivery to help prevent, because babies are born with their mother's immune system or their parents' immune system, if we want to be gender inclusive. So, we know that we need to support that baby until it's about 2 years old, when it really has its own independent immune system. So that could be one use. Again, it could be the sort of, just adding more options in our prevention toolbox. There might be somebody who says, coming in for an IV is an easy thing that I can do, and I'm good with that option. I don't like taking pills. I'm not so crazy about needles. I don't love getting shots, right? So just as, the long-acting PrEP that's given by injection, is another new option, this could be yet another new option. The thinking is that perhaps formulations could be developed that instead of being given by IV in a vein, could be given subcutaneous, which is in between the layers of your skin. And it's sort of, you usually give a sub-Q infusion in the fattier areas, like perhaps your belly or your thigh. And if we can get a combination of bNAbs at a dose that's able to be administered, people can self-administer a sub-Q dose. So maybe this becomes something that people could do on their own at home, rather than having to go into the clinic for a drip. So again, I think it's still kind of in flux. We're still thinking about how all this might work. I forgot one. One of the other options is to think about using it in collaboration with an injectable long-acting PrEP, because we know that those injectable drugs have this tail where if they're given, let's say every 6 months, the optimal dose may be around month 4 or month 5. And then it starts to sort of diminish over time as we get to the end of that 6-month period of time. So maybe maybe we could give you a bNAb to help sort of give you some extra coverage for that period of time where the tail is getting, sort of waning, right? So maybe there's some opportunity here for kind of a combination prevention and using multiple modalities together to have the best coverage. So again, I think all of this is still kind of in the hypothetical zone. We have to find that effective combination first, and then we can look at how to really optimize how to use it and how to administer it.
Thanks. Super, super important kind of big picture pieces to think about with bNAbs. Thanks again, Gail. So maybe just in the remaining minutes, we'll do something fun. And we're going to do these sort of multiple choice questions. This is all anonymous. So don't feel shy about answering. But so neutralizing antibodies to HIV are molecules produced by the body that prevent HIV from infecting cells, molecules in the body that help HIV infect human cells, molecules that don't have any purpose in the body. We'll just give it, okay. All right. I think, well, some people are still, there we go. So it looks like Gail, great job on the overview. People got that one. So maybe I'll hand it over to Daisy. Or Daisy.
Sure, I can read out the next question. We've had sensitive and resistant. So a highly sensitive virus, HIV virus, is three options: a virus that causes more severe disease in a person, an HIV strain that will be blocked from entering a human cell by a particular bNAb, or a virus that cries a lot. Highly sensitive.
So this is still a point that's a little confusing. Yeah. So again, the idea of some viruses are going to be vulnerable to the antibody, and so it's not that it's a virus that will cause more severe disease. It's that it's a virus that is more susceptible to being killed by the bNAb.
Fantastic.
And I like that the link you made to the vulnerability. One that's sensitive is one that's vulnerable to the bNAb.
Okay. Okay, so I will read one more question and then I'll kick it over to, I guess, Nandi. So achieving proof of concepts in a trial allows the experimental product to, 3 choices: get licensed and make the product available for sale. Second, continue being researched and providing more clues about efficacy. And third, it allows the product to get out of jail. Proof its innocence, I guess.
Yeah, so this is one of the really important things about the AMP study that we actually educated about right from the beginning, was that this was not a study designed to bring a product to market. This was a study that was just for the scientific purpose of proving whether this concept, this idea of using a bNAb for prevention could work. So we've tried to be really candid about that since the very beginning. And now we have proof that, in fact, the concept works. So, so now we'll be able to look at how we build on that and how we design better trials going forward.
Mm-hmm. Okay, so I think I'll hand it over to Nandi to take us through another couple of questions, and then she can kick it over to whomever she likes.
Thank you, Daisy. So the next question says, the breadth of a broadly neutralizing antibody refers to 1, how nice it smells, 2, how many virus strains it can neutralize, or 3, how well it prevents HIV. Jessica or Stacy, do we have the results yet? Thank you. So 100% agreed that it's broadly neutralizing antibodies the breadth of a broadly neutralizing antibody refers to how many virus strains it can neutralize. Next question, please. So you have two antibodies, one with high potency, and we'll call that one H, and the other with a low potency, and we'll call that one L. Which statement is true? H can knock out L, H neutralizes virus at a low dose than L, or L neutralizes virus at a low dose than H.
Right. So this is that the line graph I showed you with all the different curves, right? We want to have a BNAb that we can use a low dose, a small amount of antibody, but have the highest effect in terms of blocking. So that's the potency.
I think I'll kick it over to Ntando to read the next couple of questions.
Perfect, thank you, Nandi. And we continue with our science exam, and our principal is Gail Broder. Escape, the observation that some antibodies can escape neutralizing by a BNAb is called viral sieving, viral phishing, viral pixie.
And I think this one was put in there to stump you because I don't remember if I actually used this term in my presentation or not.
Yeah, trick question.
Yay, so most of you got it right. So if you've ever used a sieve in your kitchen, or you might know it as a strainer or a colander, right? It's that thing that looks like a bowl, but it's got holes in it so the water can drain out. That's exactly what we're talking about here. So, excuse me, a virus that is not susceptible to the antibody, and so it actually can still get through and it can still attach to your cells and cause an infection. It's just like the water going through that strainer. So it is being sieved. The sieve is catching some of the viruses and it's letting some of them through.
Great, the next question, and we see the questions and remarks keep coming, and Jim really raising a number of things, but let's go to in vitro. In vitro sensitivity tests are conducted in humans, laboratories, mice and monkeys. Which one do you think? And what Jess is saying, that this is anonymous, it's true, so please ignore my earlier remarks. There we go, Gail.
Nice. So yeah, so it's when it's in vitro, we talk about it being in the lab. That's the experiments that are in Petri dishes and test tubes. It's when it's in vivo, which is in a living being, that we start looking at whether it's in animals or in people. And so I understand that the Latin is confusing for sure.
Yeah, in vivo, in vitro. And that takes us, I think we don't have any more questions. In fact, we are reaching the hour. We do. Strong product. If we want a strong product, oops, that went away. I don't know if it's because of time.
There we go.
There we go. So if we want a strong product, a low IC80 for an antibody, is that good? Is it bad? Is it disastrous?
Oh, let's see, it's not letting me submit. Oh, there it went.
I guess you'll start with the low IC80 there, Gail.
Yeah, so remember we want to see whether, it can inhibit 80% of viruses. So, oh, people actually got it opposite. Okay, so we would want it to have a strong percentage, a high IC80, which would mean it was doing a very good job of inhibiting 80% of viruses. So people actually got it backwards.
Perfect, thank you. And that should be it with questions, and we hand it over to Stacy as we are on top of the hour.
We are on top of the hour, so I will be super quick. I think we were, again, we wanted this to be, just a place for advocates to come and just understand BNABs better, and especially thinking about the AMP results that were presented last week, and not specifically those results, but I think how we understand BNABs, a BNAB product, these different results and different data that will come from the AMP trial, but other trials of other BNAB products. So Daisy mentioned at the very beginning, which I thought was the greatest way to start this this session, and many of you weren't on yet, but she said, I think you just have to keep talking about BNABs. And so it's hard. I think we always, as advocates especially, we want something to come out of these meetings and these conversations, and that is the ultimate end goal. I think, Jim, you've hit on some really important points, because I do feel like it's BNABs are a bit of an abstract concept. And I think given that the AMP study was just a proof of concept study, it's been a little bit hard to kind of think about, okay, well, so what are we actually thinking about in terms of getting one of these products, actually out to market and part of the prevention toolkit. And so this is the beginning, I think, of those types of conversations. So really glad we got to touch on that. And I think, as we all know, the field only moves forward by, I think, science and community and advocacy issues sort of all coming together, and pushing it. So this has been really great. But again, I just want to say, Gail, thank you so much. Early morning, you were very on top of it, and thanks for facilitating us through those silly multiple choice questions. I think they really helped kind of unpack the concepts a bit more. And I think just to note for everyone that that I think as an HIV prevention community, we will be talking about BNABs, probably more together. We'll be talking about AMP as the publication comes out and as we unpack the data more. So stay tuned for all of that. And we will continue the conversation.
Yeah, there will be lots more to talk about.
Yeah, for sure.
The studies of combinations are already underway, so stay tuned.
Yeah, yeah. Great.